Welcome to the Education Outreach and Training tutorials. Today I'm going to be talking about an introduction to short read mapping, which is the foundation of next generation sequencing analysis. So this material is available under the CC BY SA 3.0 license, which means that you are open to copy and redistribute it as long as you give credit to the original creator, which is myself, unless otherwise noted. The commands, datasets, and scripts will be available afterwards for your own self-paced exploration. So I've already been introduced. I've worked in genomics for a while now, and this is my puppy, Mr. Sherlock Holmes. So a quick outline for what I'm going to be talking about today. I'm going to touch on next generation sequencing data and how it can be used for diverse data types. We're going to take a glance into transcriptional regulatory data sets and where to find them. Then we're going to map reads to the reference genome using a tool called BWA-MEM. We will call peaks and create pileup files using MaxTo. And then lastly, we're going to visualize everything we do in IGV, or Integrative Genomics Viewer. So in order to understand the data, I'm just going to walk you through how it is generated. So next generation sequencing, we start with fragments of DNA. We ligate adapters to create what's called a sequencing library. That's these purple and pink adapters. And then they go on to one of these fancy machines. This is an Illumina HiSeq X machine. And so on this machine, these short pieces of DNA with these adapters are going to ligate to what's called a flow cell. They're then going to amplify into small clusters. And then we're going to proceed with sequencing by washing over these fluorescently labeled nucleotides across the entire flow cell. So when a nucleotide matches to a given piece of DNA at a given spot, after being excited with the laser, it will emit a fluorophore and a picture will be taken. So at this spot, there's a G at this piece of DNA. This other piece of DNA has a T, a C, and an A. So that's the first position that we are sequencing within our reads. And this proceeds throughout the entire length of the read by extending and continuing to excite these fluorophores and taking these images. And by the end of it, what we have is essentially a set of images that get converted into a FASTQ file. So this FASTQ file, you may have heard of this if you've worked with this data before. It has a standardized format where there will be the read name, followed by the sequence, followed by the plus sign, and then this crazy set of characters which indicate quality scores. So it doesn't matter what your input data is, the output from these machines is very standardized. So whether you're doing DNA sequencing, ChIP sequencing, which we're going to talk about today, RNA-seq, GRO-seq, ATAC-seq, many of these assays, even the non-DNA assays, they do undergo a conversion into DNA before sequencing. So all of these different data types, we end up having the same output file. So the quality scores are ASCII encoded, which means that each individual character corresponds to a score between 0 and 40. Now those quality scores, they essentially represent the percent chance or the probability that the score or the position that base that was called is an error. So it's not a perfect process, and we will have error in this sequencing process, and it's good to know whether we have high confidence that there is a low probability of error with a high score, or if there's a very low score, that indicates a high probability of error. Obviously, that's a rapid introduction to a very complex technology, and I recommend if you're new to this technology and you're going to be using it within your research, go online, especially to YouTube. There are really pretty videos that are much better than these diagrams which I've drawn for you here. So next up, we're going to talk about transcriptional regulatory data sets and where to find them. So ENCODE, or the Encyclopedia of DNA Elements, is a large consortium which has been collecting many diverse assays over a number of different tissues and cell types for the human genome. And so these assays, they include things that will investigate how the DNA interacts with itself and how it's organized within the nucleus. It will also include things like ATAC-seq, which we're talking about today, which investigates the regions that are open and accessible for proteins to act along the DNA. ChIP-seq, which is another thing we're going to be talking about today, which can look for proteins that are interacting with the DNA, or for specific histone marks, which are essentially indicating that the DNA is wrapped around a protein in a given conformation. And then lastly, another thing we're going to be looking at today is where RNA polymerase, which is transcribing this piece of DNA and producing RNA, where it is along the DNA genome. If you are looking for data to play with within your own project, or if you're looking for more data to analyze after today's session, then I highly recommend going to this link, encodeproject.org. And if you take a look at my next slide, by going to this link and clicking on the data tab here, you can see that they have thousands of data sets over a variety of different tissues and cell types, including whole organisms, or, wow, carcasses, and then the number of different assays which they have produced. This ENCODE data set, or the ENCODE portal, you can have access to a number of different data sets through this portal, including ChIP-seq and ATAC-seq. This is what we're going to be talking about today. So I've said ChIP-seq a few times, and it's going to be important because we're going to be analyzing data that comes from it. So here's a horribly drawn illustration of what is ChIP-seq. So there are many different purposes. The diagram I'm going to show you is pretty much showing ways in which we can find the sequences of DNA that a specific protein interacts with. So this example is for a transcription factor, but anything that binds to the DNA can be similarly assayed. So the first step is that the DNA drawn here is cross-linked to the proteins. Then it is sheared, perhaps using something like sonication or a restriction enzyme. And then it's washed over a column where the protein of interest will be grabbed by an antibody attached to that column, so that these DNA protein complexes are kept within the column, and everything that does not attach to the protein is washed away. We then reverse the cross-link so that we only have our pieces of DNA that we are looking to analyze. And then we take that through the process of building our sequencing library, which I talked about before. And then that's going to be sequenced on the Illumina machine, and we're going to get out these short pieces of DNA, which we will call reads. ATAC-seq is somewhat of a similar assay, but instead of finding places where the DNA is attached to a protein, the process of ATAC-seq is that it will find regions of DNA which are open and exposed. It will then essentially pull down those regions with a specific restriction enzyme, amplify them, and then sequence them. Now, as you can see across this diagram, a review from Nature Genetics, there are many different data sets which are all giving us insight into how the DNA is wrapped or regulated. And a critical step within all of them is obviously the sequencing, but additionally the mapping. And so this is what we're going to be focusing on today, is taking this data, which we have already been produced by the ENCODE consortium, we are going to map it against the reference genome, and then we're going to visualize the output. So individually, when these sequences come off the machine, they don't have much information. The raw data, as we refer to it, or this raw unmapped FASTQ file, doesn't have much utility and is not easy to interpret. However, collectively, they can represent something useful. So for us in this ChIP-seq and ATAC-seq, we're going to be going over reference-based mapping. And the process there is that we have a reference genome, for example, the human reference genome. We can map these reads against that reference genome to see where a given read matches up to the reference genome sequence. And then we can find places where those reads are piling up or where they're aggregating in a single region along that genome. And this would be determined in enrichment. And by looking for this enrichment, we call these peaks. And if this was transcription factor ChIP-seq, then these would be regions which we would be considered to be bound by the transcription factor that we've assayed. The same process is going to be the same for polymerase ChIP-seq, which we're going to go over today, as well as ATAC-seq and H3K27 acetylation. All of these assays that we're going over today are about finding these zones of enrichment after mapping them against the reference genome. So now we're going to go into the process of mapping reads to the genome using BWA-MEM. So for those of you who want to follow along, I'm going to be showing you files and pipeline scripts which will be available for you to reuse or repurpose as you see fit after the session. I do not expect you to follow along on the command line while exploring these files, and you can always rewatch this recording and hit pause, maybe listen to slow motion, whatever makes you happy. So I'm going to go ahead and minimize this presentation, and I'm going to open up my terminal and log into the Cedar compute cluster. So I use this horrible yellow color, which some people are not fond of, but it works for me. So I'm going to make this a little bit bigger for you. SSH cedar.computecanada.ca. If you'd like to follow along, you can log in as well. Perfect. Okay, hide that side panel. Perfect. And okay, so the directory, which is going to be relevant for today's workshop, can be found here, and you should have access to that directory. So I can go change into that directory, and if you're not familiar with how the command line works, that's not something that I can teach within this session. However, there are other tutorials on Compute Canada's systems which will allow you to gain these skills. So for now, if these commands are foreign to you, if you don't know what I'm doing, then I recommend taking those other courses. So this is my workshop directory, and I'm going to take a look at the scripts directory. And in here, I have these different scripts that end in .sh. So I'm going to be walking you through one of these scripts piece by piece. But if we want to take a look at what one of these scripts looks like all together, this is how it looks. So at the top, I'm going to go through this again a lot slower, but we have some information about how much compute resources we're going to need to process this data, the account that's going to be billed to. We're going to load our necessary requirements. We have some genomes, and then we have some more variables. And then the core piece here, we are going to be mapping the reads to the genome, converting them, and calling peaks. Now, I don't expect you to follow along with what I just went through, so it has been broken down piece by piece. If you do want, you can make a temporary directory within this location to play around with after the session. I don't recommend doing it now because it might be tough to follow along. I have made such a directory, and I've named it Sherlock. So what's nice is Dr. Forness has made these scripts for us, which include the ability to download data, create a directory, process that data, all in a single script. So by modifying these scripts, these are already functional as long as you modify them to be relevant to your own account information, email address, and directory location. They should work just fine. So I've copied these scripts into the directory Sherlock. The scripts that end in underscore workshop, these are the ones that you're going to want to edit. So if I take a look at, for example, this ATAC-seq workshop script, up at the top, you can see if I was to submit this to the scheduler right now, it would not work. You'd have to replace this with your account, replace that with your email address, and change this to be your specific working directory within April 2019. Okay, so welcome to the Mellow Yellow. These scripts, when we're submitting a script to the scheduler, they're all going to need some specific information which we pass to the scheduler via these commands that start with the hashtag followed by sbatch. And if you are completely foreign to this, there are tutorials and information on the Compute Canada website about how to build these headers. So for example, here I'm going to be using 50 gigabytes of RAM. I'm going to be using 16 CPUs on a single node, and it's going to have a max time of 4 days. Again, you need to change these to be relevant to your own case if you want to reuse these scripts. The next section, we're going to be loading the necessary tools. So we're going to need to use Python. There is these two commands here which are going to install MaxTo and NumPy into your home directory under .local/bin/max2. We're going to use module load BWA, so this is a tool that's already on the Compute Canada system, and also module load SAMtools, again, a tool that's already on the system. Next, we're going to need a reference genome. So the reference genomes are going to be the things that we are mapping our short FASTQ files against. So I realize that not everyone will work in human. However, if you work in a model organism, there is a repository which has many different genomes curated by a team at McGill. So here, I'm going to be using the FASTA file of the reference genome and the BWA index of that reference genome. So what is a FASTA? So a FASTA, this is just a format, a FASTA, call it what you like, that specifies a given chromosome name, some other information, and some accession information on a line that starts with a greater than sign. And then it will have every nucleotide within that reference sequence for that chromosome. When the next chromosome appears, there will be a new header line followed by that same sequence. If you do not have a pre-built index with BWA for your genome, you're going to need to run the BWA index command. This will generate a number of files that end in .amb, .ann, .bwt, .pac, and .sa. Now, what is actually within each of these subfiles is not the focus of this presentation, and I've been doing this for about 10 years, and I can tell you I don't actually know what's in any of those files. So for me, all you really need to know is that this is an indexed representation of a long linear FASTA sequence. So this is going to allow the tools to map against an enormous reference genome, for example, billions of nucleotides, in a matter of minutes or hours. The next section of the script, I'm going to be setting some more variables. Something just briefly about setting variables like this. This is something that I highly recommend. It is going to allow you to reuse the same scripts and the same commands while changing out individual input data files or perhaps working directories. And so the reason that we've set up the variables as we have here is such that you can change your working directory to be relevant to your own system or your own path, and then everything else within the script will still be functional. So I'm setting an identifier, ATAC-seq. I'm setting a working directory, which is going to be within the April 2019. I'm calling this one data2. There's also one in there called Sherlock. You can call it whatever you like. And then this $ID is going to take this string and append it to this variable. The platform is Illumina, and we'll need that. I'll show you why later. The sample is a heart left ventricle. We're using 16 threads. And then we are going to create that working directory, and we're going to change into it. Then we set some more variables, and we're going to download some data. So the next variables we're setting are FASTQs. So this is going to be the path to the first FASTQ file, the path to the second FASTQ file. And again, you can see we're referencing these previous variables that were listed above. We're also going to set a max directory and a peaks file name, and you'll see where we use each of these variables in the commands below. Then we're going to download some data. So we got these links to these specific FASTQ files, which are compressed with a gzip compression, from the encodeproject.org. So again, if you want to download different files, you could just swap out these links for data that's relevant to your own system of interest, and then process still in the same manner. Again, I highly recommend visiting this website. There's lots of open source data for you to explore. So now into the fun part of the script. We're going to be mapping reads to the genome. So this if or fee statement is just going to be checking that the output of the command doesn't exist yet. And so this is a way that if we were to run our script and this was the only part that finished, then we wouldn't have to rerun this step when we were rerunning the script. Now digging into the core of the command, what's actually within this block here, to run BWA mem, you really only need two things. You need the index base, so this is going to be that genome index path. You also need an input FASTQ file, and if you've got paired FASTQ, then you would also give that second FASTQ. I've also added the options for threads and -m. So this is going to allow it to work in a multi-threaded fashion, which is going to increase it by the number of threads that we have available to us. Also, the -m will allow the reads to map split and secondary hits, which is not always needed, but it's an option that we're including here. Then there is this read group string, which is going to be added to each read, and it's just going to tell us something about the sample ID that we're sequencing. So this would be ATAC-seq. It's a heart ventricle, and it's been sequenced on the Illumina platform. Lastly, we are going to capture the output. This would print just a standard out. Instead, we're going to capture that within a SAM file. So the output of this is going to be, as I just said, a SAM file. And a SAM file, again, is a standardized format which has sequence. Sorry. It has some sequence information about the different chromosomes or contigs from the reference file. It'll have information about the program which you mapped it with, for example, BWA. And it'll have the read group information, which we actually do have because we put it onto our mapping command. I'm going to go ahead and show you what one of those SAM files looks like. So we will take a look at Sherlock, and we will go into ATAC-seq, lift this up, and we can go ahead and take a look at this heart ventricle.sam. So again, I've already run those scripts. Those scripts are going to produce this set of files. And if I take a look at this SAM file, you can see this is information about the reference genome which I mapped it against. Then there is information about the command that I run. This is the command that I run. And then for each read, there's going to be information about the chromosome and the position which it was mapped to, as well as a number of other flags and columns, which for the sake of time here, I'm not going to go into, but if you'd like to learn more about them, this is a good place where you can learn about the SAM file format at this link here. So once we have our SAM file, that's just a standard text file. That text file is not going to be read by downstream tools. Instead, for the sake of space and compression and access to different parts of that file rapidly, we're going to use a suite called SAM tools in order to convert this file into a BAM file and sort it. So in this command here, what you can see is I am running SAM tools view, which is going to take an input SAM, and it's going to leave an uncompressed output with a number of threads. So this is going to be multi-threading the SAM tools command, and it's going to take our input SAM file. Then I'm linking this with the pipe. So instead of saving an intermediate BAM file, I'm just piping this directly into the SAM tools sort command, which will then operate with multi-threads, and it will output our final BAM file. Lastly, I'm going to index that BAM file, and this is going to be necessary for visualization later. So we'll produce a .bam.bai file next to the BAM file, which is needed for these downstream tools. There's an easier version of SAM tools if you don't want to link the commands as I did there, which saves time and space. But if you're just learning for the first time how to use these tools, then you can use a more simple approach, which would be first to run SAM tools view on the BAM file, on the SAM file to create a BAM, then SAM tools sort on that BAM file to create a sorted BAM, and lastly SAM tools index on that sorted BAM. So once we have our sorted BAM file, we can take that and plug it into downstream applications. So because we are working with this enrichment style data, whether it's ATAC-seq, Pol2, or histone 3, H3K27 acetylation? Yeah. Then what we're going to do is we're going to be looking for regions of the genome that these reads are mapping and piling up in an enrichment. And we're going to use that tool called MaxTo in order to perform this. So again, the final component of this pipeline, again, if we have our output, then there's no need to run this. But if we don't, then what we're going to do is we're going to call that MaxTo command, which we put at the top of our script. MaxTo has many options. One of them is call peak, and we're going to give it an input BAM file. We're going to name it by the sample ID. We have a threshold for significance cutoff. We have an output directory where these results are going to go. And then lastly, I want to store a bed graph, which is normalized per million reads mapped, and that's going to help with visualization after the fact. For some types of enrichment, for example, the ChIP-seq, there is a control which we can set with the -C option. And if you explore those other scripts, you'll see that -C option for the control. So the output from MaxTo, it's going to give us several different files. The ones that we really need to pay attention to are this treat pileup.bedgraph, the summits.bed, and the peaks.narrowpeak. And I'm going to visualize these files in just a moment. You'll see what they actually are. So for visualization, I like to use the OSX fuse or SSHFS in order to connect my computer to the Cedar file system. If you don't have it installed, you just Google OSX fuse, and there's a very straightforward approach. It works for Linux with SSHFS and with Mac OSX fuse. There's also a tutorial, 2-minute learn-along, describing this process from Compute Canada, and if someone's really interested in this, then we can dig that up after the presentation. So the command that I'm going to run is I'm going to run, once I have it installed, SSHFS. I'm going to use my login information, and then I'm going to use a specific directory which has my data, and I'm going to link it to a directory on my own machine, which I just call portal. It's going to then prompt me for my password, and then it will mount my computer so that it's exposed to the Cedar compute cluster. So I'm going to go ahead and do that now. So I can make a directory called, we'll call it portal workshop. So I'll do a SSHFS cedar.computecanada.ca scratch Richmond P training April 2019, and we're going to link that to portal workshop. Ask for my password. So now, if I look at portal workshop, it's exposed that same set of data as if I was just on the cluster itself. So in this window, I am on Cedar, logged in, and this is what I see. And here, I am not on Cedar, I'm just on my own laptop, and I can see these different directories. I highly recommend using this for data visualization so that you don't have to bring files down onto your own system, especially since sometimes these files will be dozens or hundreds of gigabytes large. So next I'm going to open IGV. If you don't have it downloaded, then you can download it here after the webinar. IGV, or Integrative Genomics Viewer, will open up in a window that looks like this. So here we can select our reference genome. If you don't have the proper reference genome, HG38 is what this data is mapped against, then you can click, and HG38, you can see this is going to be each of the chromosomes, and we can now load files that we've mapped into this space. I've got a little walkthrough on how to find it if it isn't already in your list of optional drop-downs. You can see that you can find it by clicking on this More button here. So then we're going to go, we want to load our files, so we'll go File, Load from File. So we're here, File, Load from File. If I go to my home, you can see I've got this OXS Fuse volume. And this is now, again, I'm still just on my computer, but I'm able to see the data which is on the cluster. So I've got a few different data sets here. ATAC-seq is one of them. So we're first going to load this ATAC-seq. So if I actually go back here, you can just take a look. ATAC-seq, we're going to want to select the BAM file. So these are going to be the actual reads. And again, this is saving us from downloading the data onto our computer. And we're going to also select from the MaxTo directory, we will select the narrow peak. So I can open both these files in IGV. And you can see we've got these peaks throughout the genome where the ATAC-seq data is piling up. And we've also got our BAM file now loaded here. We can go ahead and rename this track if we like. We call this ATAC-seq. Right-click and rename ATAC-seq. And I think I've pasted, yep, so again, we can load the BAM file and the narrow peak. And we have a region around the gene NEBL where we will take a look at this data. So we can paste that region into there. Okay, so as you can see, we're still zoomed out quite a bit, right? We are about 2.2 megabases is this entire window. So that's too far zoomed out to actually visualize our reads. But you can see we have lots of different ATAC-seq peaks. And we also have down here at the bottom different genes. So if we select one of these, let's select a peak within a promoter. Okay. So if we zoom in, we're at 87 kilobases. You can zoom by clicking and holding and dragging here. We'll zoom in a little bit more. Hopefully it doesn't break. It's still thinking. And there we are. So now what you see is that the ATAC-seq reads are piling up at the transcription initiation site of this gene DNA JC1. So we can continue to zoom in, and you can see that these reads are going to be the individual sequences that were coming off of the machine. If we continue to zoom in, you can see some of these reads have soft clipping happening, which means that there are mismatches. And this is coming from the fact that the pipeline we are running here does not include some quality steps, which are probably recommended. If you're interested in those quality steps, including adapter removal, then you can talk to us afterwards and we can show you how that process works. So we've got our reads piling up at this promoter. If we like, we can select this peak file here, and we can scroll through these different regions. What's interesting here is that we're seeing some peaks piling up, and there is no gene at this location. So this could be representing a region of open DNA that's perhaps an enhancer. So because we've got other data which we can load, we can go ahead and load the H3K27 acetylation. Now we know that this data is representing active enhancer regions. So if we see some evidence of H3K27 acetylation at this position, perhaps we are looking at an enhancer. Again, when we load this data, it's going to take a minute to think. And believe it or not, yes, this data is showing that this region, where there's no gene, does have quite a bit of H3K27 acetylation. So what you can also see in the H3K27 acetylation data is a bit of periodicity, which matches to the nucleosomes along the DNA. The last file that we're going to load here is going to be the polymerase II ChIP-seq data. If we take a look at the polymerase II, we can look at our left ventricle, and we will also load the peak data. And so now we will see if the RNA polymerase is also overlapping this non-coding position. And you can see it is, so we have evidence from these three different data types that this region of the genome where there is no annotated gene has a peak for ATAC-seq, meaning the DNA region is open. There is a peak for histone 3 H3K27 acetylation, and there is also a peak for RNA polymerase II, meaning that this region is being actively transcribed. Now, what else do we have? The last thing is that one of the limitations from this data visualization is that when we zoom out, you can see we are limited in how far we can zoom out. For example, we can only see about 30 to 50 kb. And one way in which we can just capture this coverage track without needing to visualize all these individually red and blue reads is by converting our bed graph data into bigwig. Oh, go away, Slack. Sorry about that. So there is a script for this, and if you're interested in that, I can show it later, but I think the most useful thing we can do now is kind of open it to questions and comments, and happy to field those as they come in. I will monitor. Perhaps in the video, if you'd like, you can unmute your microphone and ask a question, or if there's something from the Etherpad, doesn't look like it was really being used. Was anyone on the Etherpad?
4 people.
4 people? Oh, I guess I don't have the up-to-date one. And other than that, thank you for listening and participating. I encourage you to attempt to run these scripts, which are again deposited here, and repurpose them so that you can use them in your own data analysis. That's Philip Richmond and Oriel Forness, and thank you for listening.